I'm also trying to get the award for the cutest study animals of any of the talks. Today I'd like to talk about really just the importance of taxonomy, good taxonomy for conservation biology, just focusing on a couple of case studies that I've been working on bandicoots and gliders. So why is taxonomy important? Aside from underpinning really all comparative genetic, ecological and behavioural studies, taxonomy provides the basic units for conservation management, the species and subspecies, and also higher levels of taxonomy. The implications of uncertainty in taxonomy can be quite profound. First, there can be unrecognised endangered species which may go extinct before they're even recognised, and an example of that which hasn't fortunately gone extinct is a new tuatara species which was found relatively recently in New Zealand. Resources may be wasted on incorrectly identified species and subspecies. And one case that I did do some work on in the past is Sminthopsis griseoventa boulangerensis on Boulanger Island, which has finally been removed from the EPBC list. And aside from that, its translocation or genetic enhancement programs really need taxonomic information. There can be unintended consequences by translocating different species together or different subspecies together, which may lead to reproductive fitness loss or even the potential for new hybrid species. So just this talk's focusing on mammals, and you would think that we actually know the taxonomy of mammals pretty well. But new species of mammals are turning up all the time. And just a couple of examples. Here in the recently published rock wallabies, a new genus of bats and new species, and also last year a new antechinus species was described. There are also many ongoing conservation issues associated with subspecies recognition. And for the rest of the talk, I'd just like to focus on two case studies, the southern brown bandicoot and sugar gliders. Before I do go on, though, I think I should define what I mean by subspecies. And I'm going to use a definition that was proposed back in the early 1990s by two separate papers, John Avies and Ball, and Steve O'Brien and Ernst Mayr. And nicely summarised by Dick Frankham, subspecies are groupings of populations within species that share a unique geographic range or habitat and are distinguished from other subdivisions of the species by multiple independent genetically based traits. So quite a mouthful, but what they introduced was the concept of genetics in subspecies and the potential for different criteria, genetic criteria, to be used to distinguish subspecies or to test subspecies boundaries. These can be the gene trees, where we have different populations in monophyly, either reciprocal monophyly or paraphyly. So population B is in monophyly. Its sequences are most closely related to sequences in population A than other sequences in population A are. So those kinds of patterns in gene trees, and this is another one, a haplotype network, haplotypes just being different sequence variants, circles indicating the frequency of haplotypes, and where you get fixed haplotype differences, it's an indication of some level of population isolation and the potential for different subspecies to be present. So, getting back to case study one, the southern brown bandicoot, there's been numerous taxonomic changes over the years, and I'm just going to utilise the recent paper by David Paul, which recognised 5 subspecies distributed across southern Australia. They have very highly fragmented distributions. The subspecies I saw it on, Abesilis is nationally endangered under the EPBC Act, and recently there have been doubts cast on the subspecific status of the Tasmanian subspecies Affinis and a subspecies which is off the Eyre Peninsula, Nauticus. And this is based on some genetics work done back in the early 1990s, and more recently DNA sequencing work done by Pope and Zenger and colleagues. And this has led to a recent proposal to remove the subspecies status of Isoodon abessilus abessilus. Essentially based on a redistributed range where there are very secure populations, particularly in Tasmania. So together with a whole bunch of colleagues from across Australia, we've decided to test this notion of subspecies boundaries using some new genetic techniques or new genetic markers. We obtained around 150 samples from across Australia, including the species Auratus and also Macrourus, which are known to be closely related to abessilus. And we obtained sequence data from 5 genes, 2 mitochondrial and 3 nuclear genes, analysed these data using a combination of phylogenetic and haplotype network analyses to get an idea of whether there has been population isolation. But importantly, we included around 20 samples from Tasmania, and previous genetics work, particularly the DNA sequencing work, had virtually no samples from Tasmania. So you throw all the data together, produce these gene trees, and the standout from just looking at the mitochondrial data is that there's 2 main phylogenetic groups, one associated with the species Macrourus, and a second group associated with abessilus and Auratus. Notably, considerable intraspecific genetic variation within each of these different groups. So I'm just going to focus on this big group, depicted in this way, showing the different colours associated with different subspecies and their locations. Genetic divergence levels in this marker, in these markers, was around 4% or less than 4%. But what's quite evident in this tree is that there's a major east-west split. So we have a lineage associated with abessilus abessilus and affinis, and a second major lineage associated with the Western Australian subspecies Fusiventa and the Mount Lofty Ranges, Kangaroo Island populations in yellow here, and the other species, the golden bandicoot Auratus, mixed up within this group. And there's a separate lineage associated with the North Queensland subspecies Peninsulae. So what's also evident is that the Tasmanian subspecies forms a monophyletic group, indicative of long-term isolation of this population. Its divergence from the mainland being around 2.3%, and if you like rough molecular clocks, that might equate to 1 million years of potential isolation, or at least coalescence of the genes in this gene tree. It's also evident that the Lofty Ranges population really shouldn't be considered part of the subspecies abessilus abessilus. So we propose from this mitochondrial analysis that it supports the idea that abessilus abessilus is restricted to the southeast eastern part of Australia. Now, if you look at nuclear markers, this is slightly complicated, these haplotype networks, size indicating frequency. These nuclear markers that we use are quite conservative and are not very good at even separating out the different species. So you can see Macrourus shares a haplotype with RAG1 and similarly with BRCA1. But what it does show is that there is evidence of some fixed haplotype differences between the Mount Lofty Ranges population. If I just highlight those with a square. These haplotypes are unique just to the Mount Lofty Ranges, Kangaroo Island and Nauticus off the coast. And this pattern is repeated in each of the markers, with the exception that it goes with Fusiventa in one of these haplotype networks. Again, indicative of some level of population isolation, and certainly it's not supporting a grouping with Abesilis in the southeast of Australia. That subspecies shares identical haplotypes with the Tasmanian subspecies. So in summary, these data, the mitochondrial data, support the distinction of Affinis from the mainland Abesilis. Okay. Mitochondrial DNA and nuclear gene data show that Kangaroo Island populations, Mount Lofty Ranges and Franklin Islands and Francis Island populations are genetically distinct from Abesilis and shouldn't be recognised as being part of the same subspecies. It essentially implies that that subspecies is restricted to the southeast regions of South Australia, Victoria and New South Wales, a significant reduction in the population size of this taxon. So our recommendations is that it should maintain its endangered status and separate management should also be undertaken for the South Australian populations. And I just want to finish off by just quickly going through another example system. This is some work I'm doing with Sue Carthew up in Darwin on sugar gliders. And it was really based around some initial genetic analyses that showed that the sugar gliders in the Northern Territory, which was supposed to be related to Ptoris brevicets, turned out to be something a little bit different. And so we've been looking at the question of what glider species exist in that part of Australia. Excuse me, I'll just take a drink. So Sue's conducted a big trapping study of this region, collecting about 20 sites. And we've then conducted mitochondrial DNA, and we're in the future conducting some more nuclear gene analyses of these populations. But what was evident from the mitochondrial data is that the Northern Territory sugar gliders are not related to Ptoris breviceps. In fact, they're much more closely related to squirrel gliders. And there's also evidence for some other distinctive genetic lineages in Northern Australia. So if we just home in on that part of the tree, there was evidence for this one big group with a very wide distribution across Northern Territory, but also a very different genetic lineage was found in the Kimberley region, and even a sample that was related to a mahogany glider was found in a population near Darwin. Each of these show quite high divergence levels. Nuclear data at the moment, this is very preliminary, but it also shows the distinction of each of these different populations, the Northern Territory lineage, from other Ptorid gliders. We're also collecting morphology data, and just a snapshot of this. It's essentially a very small glider, much less than a sugar glider in size, but it's got quite a large head. And these are preliminary data, shows that there are probably some very distinctive morphological differences for this glider species from other gliders. Lots more work is needed, and this is happening over the next few years, we hope. But just to summarise, it's likely that there's 2 new species of gliders up in the Northern Territory and Kimberley region, and of course then they're of very uncertain conservation status. There's also potentially a new population of mahogany gliders. It's an endangered taxon in Queensland. It has a slightly different sequence in Darwin. Perhaps it's an escaped pet, we don't really know. But gliders have very specific habitat requirements. They need nesting hollows in trees and are very tree dependent and potentially vulnerable to controlled burning practices that are inappropriate. There's a potential link between a big decline in mammals in the Northern Territory, and so this species is in bad need of assessment. I'd like to thank a range of colleagues from across Australia who've contributed to this study, and thanks very much for listening.
Thank you, Steve. We won't have time for questions, I'm sorry. We'll
Yep.
have to move straight on to Rick Davies.